Hi everyone, today we're going to learn cell and nuclear division. First of all, what is the meaning of cell division? Cell division is referring to the existing cell undergoing division to form new cells. Now, the next question will be, what is the purpose for cell to undergoing division? Simply, it means for reproduction and growth. As in unicellular organisms such as bacteria, the purpose of cell division is for them to form a new bacteria cell. That is for the purpose of reproduction. So in unicellular organisms such as bacteria, the process of cell division is known as binary fission. While for multicellular organisms such as human, the purpose of cell division is for the development and growth. As in human, we have a lot of cells in our body. When each body cell undergoing cell division, it allows us to grow taller. Other than this, cell division also important for repair and regeneration. For example, sometimes when we have cell injury or body injury due to accident, so the injury part, the wound area, the cell will be undergoing division so they can form a new cell to replace the old or damaged body cell. So there are two types of cell division in multicellular organism. There is mitosis and meiosis, which we will discuss both of them in this topic. And remember one thing, mitosis and meiosis is not found in unicellular organisms such as bacteria. The cell division can also further divided into cytokinesis and karyokinesis. What is cytokinesis and karyokinesis? Cyto means cell. So cytokinesis means cell division, means dividing of the whole cell, including the nucleus. Wow, karyokinesis. Karyo means nucleus. So karyokinesis simply referring to the division of nucleus only, excluding division of cytoplasm. So cytokinesis including the division of whole cell, referring to the cytoplasm, while karyokinesis is only division of the nucleus, referring to the chromosome DNA found in the nucleus. First of all, as we know, human's DNA is very long. So for this length of DNA, how can it be stored in small place in the nucleus? So as you can see, this is human DNA, for example. In fact, the human DNA is wrapped around a type of protein called histone protein. So when DNA is wrapped around in this histone protein, and then the histone protein again packed together, they form chromatin. This chromatin again will condense to form into a condensed structure. We call them chromosome. This is a picture of chromosome. In fact, in one chromosome, it has a lot of DNA together with histone protein condensed together. Before we talk about cell division, we need to talk about cell cycle. Cell cycle is a complete mechanism talking about how cells get ready to prepare for their division, and then the cell going into the steps of division and then to form a new cell. Just like in marriage also. In marriage, you also need to get ready, right? You need to prepare, to work hard, to get the money, to go for some photo shooting, and then go into the actual wedding day, and so on. So it's the same thing. Cell cycle is a complete process. How cell to prepare for the division, cell going into the division, and then cell start to divide to form new cell. The cell cycle consists of two phases, and it can be further divided into four components. First of all, the cell cycle have a number of universal trends. The first one, the DNA packed into chromosome must be replicated, which means the chromosome must be replicated to double its genetic material. What is the purpose of doubling its genetic material? Or you can say doubling its DNA number amount. The purpose is so that equal amount of DNA genetic material can be distributed to each newly formed cell. Second, the copy contents of the cell must migrate to opposite end of the cell, which means is referring to the chromosome. The chromosome must migrate to the end of the cell. After it migrate to the end of the cell, then only the cell will be physically split into two separate cells. This is a general idea of a complete cell cycle. Now, as I said, cell cycle have two major phases. The first one is called interphase, and the interphase can be further divided into G1, or we also know as growth 1, S or synthesis phase, and G2 phase, which is called growth 2. After completing the interphase, then it will proceed to the division phase, which is the actual process of cell division called M phase. So as you can see, the cell cycle consists of two major components, which is the interphase. Interphase consists of three components, G1, S, and G2. When cell moving through the G1, S, and G2, then it means the preparation process is ready. After it done the preparation process, then only go for the actual cycle, actual mechanism, which is the M phase, the division phase. So you will notice that the preparation process is always takes longer than the actual process. Same like our study, you have to study for 4 years, 5 years to complete your whole degree. Then only you go on the stage to take your graduation score, which just take maybe about 5 seconds on the stage during the convocation, right? So it's the same process here. The preparation is always longer than the actual one. So in the cell cycle, the interphase itself is a preparation phase. So bear in mind that you cannot say during the interphase the cell is at rest. No, that is wrong. During the interphase, the cell did not rest. In fact, the cell is very busy in preparation. What kind of preparation the cell done? For example, during the interphase, cell is preparing to synthesize enzyme, synthesize protein that require for cell division. Other than this, during interphase, cell also double its genetic material so that the equal amount of genetic material can be distributed to the new cell. So it's a very busy phase. It's a preparation phase for the cell during interphase. After completing the interphase, the cell will moving into the division phase. At this point, the chromosome will be divided and moving to the opposite end so that the cytoplasm will be split to form new cell. This is an overview what happening in G1, S, and G2. And then after completing the G2, the cell will going into the division phase, which is mitosis, have 4 phases. Of the 4 phases of mitosis, prophase, metaphase, anaphase, telophase, in fact is a division of nucleus, or we call karyokinesis. When the chromosome is complete in division, then the cell are going to divide the cytoplasm, which is the cytokinesis. Now, let's look at the detailed process happening each phase. First of all, we start with G1, or we call gap 1 or growth 1 phase. In the G1 phase, the cell is growth, and the growth is to prepare for the DNA replication and also for some of the intercellular components such as chromosome such as the centrosome, which is the one that produces the spindle fiber so that they can get ready for cell division. At this point, what is happening is at the G1, the cell also active in synthesizing enzyme protein, which is required for cell division. So G1 phase is the longest phase. Some of the cells, they are not yet ready for division. They're not going to enter into G1. In fact, they will be exit the cell cycle, go for G0 phase. G0 phase is the non-dividing phase, which the cell still not ready for cell division. What's happening in G0 phase? G0 phase is a quiet phase and is a not dividing phase. For example, the liver cell, the kidney cell in our body, which is not ready for division, they will be present in a G0 phase. Sometimes, when the environment is lacking of some growth factor or nutrient needed, the cell will also push themselves to G0 phase. Wait for the environment is favorable or you have sufficient amount of nutrient, then only they go for dividing. So, most of the body cells in our body, they are in G0 phase when they are not prepared for division. When the cell is ready, or when for example, when you have some injury and so on, the cell is ready to go into division phase. So what happens is they'll leave the G0 phase and enter into the one phase. So in conclusion, G1, cell is active in synthesizing cell is active in synthesizing enzyme and also protein. And at this point, cell is also accumulating energy, prepare the energy for next cycle. S phase or synthesis phase. S phase is purely for DNA duplication, which means the cell is going to double its genetic material. So this S phase is purely only for DNA synthesis. What is happening is, imagine inside the cell you have, for example, inside this cell is having 1000 copies of DNA. During the S phase, the 1000 copy of DNA will be double, become 2000 copy. Why? This is to ensure that the newly formed cell receive equal amount of DNA. So as you can see, this is a piece of chromosome. In this piece of chromosome, it has 1000 copies of DNA. Okay, imagine. Now, during the S phase, this 1000 copy of DNA, it will double. Now it become 2000. When it become 2000, the whole piece of this DNA become like fatter. So what is happening is it breaks at the center, become the picture as you see here. So in this picture, it is still one chromosome. This is one chromosome with 1000 copies of DNA. Now, when it becomes 2000 copies of DNA, in this picture here, it's still one chromosome, but this one chromosome is having 2000 copies of DNA now, joined together at the center, which is centromere. So that later on, when come to the division phase, this is the result of the division phase, the M phase. This is one chromosome, this is the other chromosome. This chromosome have 1000 copies of DNA. This chromosome also have 1000 copies of DNA. So do you see or not? If the cell without synthesis phase, this is 1000 copy of chromosome. Now imagine if without synthesis phase, this 1000 copy of chromosome will be in 500 in each. Later on, when you go to the division, this is 500 copy. This is 500 copy DNA. So the genetic material will become half. So do you see the significance of this synthesis phase? Without the synthesis phase, the newly formed cell will not be able to receive equal amount of DNA, genetic material, as the parent cell. So in conclusion, the S phase is purely to duplicate the genetic material, the DNA. After completing the S phase, the cell will proceed for the G2 phase. G2 phase is shorter compared to G1, and in the G2 phase, the mitochondria and also chloroplast in the cell will be duplicated, and the tubulin is the spindle fiber which is needed to split the chromosome. At that time, it is ready to be synthesized, and then the chromosome will start to get ready to condense, and the centrioles also replicated. So after completing the G2, now the cell proceeds to the M phase. The M phase consists of 4 phases in the mitosis, which is prophase, metaphase, anaphase, and telophase. After completing the 4 phases, then the cell will proceed for cytoplasmic division, which is cytokinesis, which we'll explain later. This picture is showing the overall process of cell division. Here is the interphase, and here is the M phase or the division phase, and this is another new cycle of cell division. So I want you to take note on the chromosome number and also the DNA gene amount in the cell. So at the beginning, this is before the S phase, before the DNA replication. What is happening is, if you look at this cell, so for example, this cell is having 2 pieces of chromosome. One piece is in red color, while the other piece is in black color. So if I ask you, what is the number of chromosome at this stage? G1, the number of chromosome is 2. So the diploid chromosome number, or the 2n, is equals to 2. After S phase, the DNA is duplicated. The DNA number is double. Now, when the number of DNA is double, each chromatic is joined together at the centromere to form chromosome. So here, what is the chromosome number at this G2? The chromosome number at G2 still 2. Why? The diploid chromosome number 2n is still 2. Why? Because here you seeing 4, right? But actually the one in red actually is one chromosome, but they have two pieces of chromatic joined together at the centromere to form one chromosome. So this is one chromosome, this is the other one chromosome, and you see two pieces of chromatic joined together at the centromere. So this is one chromosome, this is the other chromosome. So the chromosome number still 2. The 2n still equals to 2. But then the DNA amount is already double. So here, the red chromosome is having 1000 DNA. The black chromosome is also have 1000 copies of DNA. So total is 2000 copies of DNA. After the S phase, the DNA amount is 4000 copy. Each chromatic is having 1000. 1000. This one 1000. This one 1000. So total 4000 copies of DNA. Okay, so if you look at here, the DNA amount is double, understand? But then the DNA amount is double, but then the chromosome number remain the same. So after undergoing the mitosis process divide each chromatic in the chromosome. So now the daughter cell form at the end, still gaining two pieces of DNA. Two pieces of chromosome, one in red and one in black, same like the original one, and each pieces representing 1000 chromosome. So the daughter cell forms here 1000 copies of red DNA, 1000 copies of black DNA. So this is showing that how mitosis process maintain the number of DNA and also chromosome in the daughter cell as the parent cell. Now let's look at the detailed process of mitosis. So the mitosis process happens in a diploid cell. So the function of mitosis is to maintain the number of chromosome or the number of DNA in the daughter cell same as the parent cell. So if the parent cell is 2n, then the daughter cell will be 2n. If the parent cell is n, haploid, then the daughter cell form will be haploid. So mitosis happens in all the body cells except gamete. Gamete is undergoing meiosis. What is the role of mitosis? The role of mitosis is to maintain the chromosome number in the daughter cell as the parent cell. So it is important in growth and regeneration and also tissue repair. Overall, the mitosis is divided into 4 phases: prophase, metaphase, anaphase, and telophase. At the end of the process, the telophase, the two daughter cells are formed, and they are genetically identical to one another, and they also genetically identical to the parent cell. Bear in mind that this prophase, metaphase, anaphase, and telophase is referring to the division of genetic material, chromosome only. It doesn't refer to the division of cytoplasm. So that means at the end of telophase, it need to undergo division of cytoplasm, which is called cytokinesis. This is just a revision on the interphase. So interphase is a preparation phase. So during the interphase, what is happening is you have a phase called the G1, S, and G2. So as G1 is the preparation synthesis of a lot of energy, the cell is synthesizing protein, and then in the G2, the cell is replicating the mitochondria and chloroplast if they have. And importantly, in the S phase, the DNA number is double. So this is before S phase. This is one chromosome. So inside the chromosome, let's say they have 1000 copies of DNA. So after the S phase, now it double the chromosome number from 1000 to 2000 copy. So each chromatic, this is one chromatic, this is the other chromatic. Each chromatic is having 1000 copy of DNA, and each chromatic is joined together at the centromere. So now this is a one chromosome. Understand? So in the first phase of this mitosis is prophase. During the prophase, what is happening is the chromatin start to condense to become chromosome. So at this stage, the chromosome become visible. The nucleus and nuclear membrane start to disappear. Why? Because so that the chromosome can scatter in the cytoplasm to be aligned by the spindle fiber. While nucleus disappear, if you remember in chapter 1 I told you, nucleus in fact is formed by integration of chromatin. Now the chromatin is condensed to form chromosome, so definitely nucleus will be disappeared. And the centrioles start to move to the opposite end. The spindle fiber will attach to the kinetochore in the centromere, which is found in the chromosome, so that they can start to align the chromosome. Now it proceeds to the metaphase. In the metaphase, the spindle fiber attached to the kinetochore found at the centromere of chromosome. At here, the chromosome will be aligned at the center. This one we call as a metaphase plate. So the unique feature of metaphase is the chromosome is aligned at the metaphase plate. Look carefully, it is the chromosome that align at the metaphase plate. It is different from what you see in later during meiosis. During the anaphase, the chromosome is separate. Each chromatic is moving to opposite end. This is pulling by the depolymerization, or which means shortening of spindle fiber. Microtubule meaning the spindle fiber. And look carefully, the migration, each chromatic, or you can say each chromosome is moving to the opposite end, leading by their centromere. During the telophase, the chromosome is arrived at the opposite end. At this point, the nuclear membrane and nuclei reappear. And then the spindle fiber will disperse. Now it proceeds for cytokinesis. Cytokinesis means division of cytoplasm. So after telophase, you proceed for division of cytoplasm. How to achieve division of cytoplasm? How to achieve cytokinesis? In the animal cell, cytokinesis is featured by the formation of this cleavage furrow. What is happening is you have found a contractile ring. This contractile ring is formed by microtubule. So the microtubule form a contractile ring, pinch the animal cell at the center. At the end, it breaks the animal cell at all into two daughter cells. However, in the plant cell, since plant cell have this cell wall, so it is unable for them to pinch together into two by the contractile ring. So in the plant cell is featured by the formation of vesicle. This vesicle will form at the middle, the center of this cytoplasm. So a lot of small vesicles will form at the center, and these all vesicles fuse together. At the end, they will break the plant cell together by forming a layer of new cell wall. So this is how cytokinesis happens in the plant cell. This picture showing the observation of each stage in mitosis. Same to this one. This is a mitosis happen in the onion cell. So by looking at the comparison, the differences between the plant cell and animal cell. In the animal cell, it involves centrioles, but mitosis in plant cell doesn't involve centrioles. What is the role of centrioles? Centrioles is to produce spindle fiber to attach the chromosome, divide the chromosome to opposite end. However, plant cell doesn't have centrioles. It doesn't mean they cannot form spindle fiber. The spindle fiber of plant cell is formed by cytoplasm. And there's a structure called ester. What is ester? If you look at picture, this is the ester. This ester is the opposite end from this pulling of spindle fiber. The role opposite end is to attach the spindle fiber firmly, to attach the centrioles firmly to the cytoplasm, so that they have enough strength to pull the chromosome to opposite end. But however, in plant cell, there's no ester, because plant cell mitosis doesn't involve centriole. While in a plant cell, the cytokinesis is achieved via cleavage furrow, while the cytokinesis of plant cell, this is in animal cell, while the cytokinesis of plant cell achieved via formation of cell plate. In animal cell, it occurs in all the tissue, but then in plant cell, mitosis only occurs in meristem, which found at the tip of shoot and also tip of root, which means other part of plant cell is unable to undergo mitosis. What is the significance of mitosis? This is very important. The role of mitosis is to form two daughter cells, and the two daughter cells is having same chromosome number, same DNA amount, same genetic material as the parent cell, and it also allow genetic stability within the population, which means that the daughter cell form is having genetically identical to the parent cell, exactly 100% same. There is no variation. And mitosis is the basic of asexual reproduction. Plants are using or some of the animal, these prokaryotic cell, they use mitosis for asexual reproduction. And sometimes when you have tissue injury, you breaking, or maybe for example in human you have bone fracture, so the mitosis process allow repair and regeneration of identical new daughter cell. So we have already known that the mitosis process is important to producing new cell, identical daughter cell. So that is important for growth, repair, and also regeneration. But have you ever wondered why the cell know automatically when should them undergo mitosis, and how the cell to ensure that the product, which is the daughter cell, is exactly identical to the parent cell, has no error. So that's the importance of this control cell cycle. This control cell cycle is having the cell checkpoint. It allows the cell to ensure the quality of product at each specific phases, G1, G2, and also M phase. It have a three specific checkpoint. These all three checkpoints to ensure that the daughter cell form is exactly correct, and to ensure that the daughter cell have the sufficient ability to complete the task, which is the mitosis, the cell division. So this checkpoint is to ensure the daughter cell has no error and is identical to the parent cell. So you can see this is a process of cell cycle consists of G1, S, G2, and also M phase. So this is a dividing cell. When the dividing cell undergo the G1 in preparation of energy, synthesizing protein, all the while when until the somewhere in the G1 phase, then you're facing the G1 checkpoint. The G1 checkpoint will check whether the cell is big enough or not. Why is important? If the cell is big enough, then only you have sufficient content in the cytoplasm for division. And also, is the environment is favorable or not? A favorable environment is referring to the sufficient amount of nutrient, enough ATP, and so that the cell can have sufficient ATP and nutrient to complete the process. If everything is okay, then the cell proceed through the G1 checkpoint, proceed to G2. At G2 checkpoint, again you check whether is it all the DNA is replicated. So to ensure the daughter cell have sufficient amount, enough genetic material identical to the parent cell when it undergo mitosis. You also check whether the cell is big enough or not, and also whether the environment is favorable. For example, has sufficient amount of ATP for division. If the cell is not fulfilling one of the criteria, you'll be arrested at this checkpoint. If everything is fulfilled, then you're passing through the G2 checkpoint, proceed to the M phase checkpoint. The M phase checkpoint will be at early of anaphase. It checks whether all the chromosomes align at the spindle. If everything is okay, then the cell will proceed through this checkpoint, undergo anaphase, telophase, and cytokinesis to complete the division process. If any one of the checkpoint is not fulfilled, then the cell will be arrested at this particular checkpoint. So this is also to ensure that the cell form is normal. So for example, like cancer, the cancer cell is due to the cell doesn't fulfill the checkpoint, and however the error cell still allowed to pass through the checkpoint, thus it produces abnormal cell, for example like cancer. So this is showing how important it is, the checkpoint, the control cell cycle. The uncontrolled cell cycle will be leading to abnormal growth. That is what we call as a cancer. However, our body cell have own mechanism to destroy the abnormal uncontrolled division cell. However, the cancer cell itself also has some method to overcome the mechanism in the body to prevent them to continue to grow. So there is a battle between the human mechanism and the cancer. Now let's proceed to meiosis. Meiosis,
occurs only in reproductive cell or in gamete in the human body. So this meiosis consists of 2 division phase instead of 1 as you observe in mitosis. So the product of meiosis is produced 4 haploid cell, and the parent cell initially is diploid. So at the end of meiosis, you produce 4 haploid cell, which is N, which means the product of cell, the chromosome number is already half from the parent cell. So what is the importance of this meiosis? This meiosis is to ensure that the next generation will have a diploid chromosome number. Why? Imagine if the gamete form is not haploid, the gamete form is diploid. So now a diploid ovum fertilized with a diploid sperm, it will form a 4N zygote. So this is already double the amount in the parent. So you see or not? So the chromosome number in the next generation form is double. It's impossible. So that's why the meiosis is to half the chromosome number in the gamete. So makes the ovum and the sperm is only half the chromosome number. So that when the haploid ovum fertilized with a haploid sperm, only you reform a diploid zygote. That's a role of meiosis. And also meiosis contribute to variation. This is not found in mitosis. Mitosis cannot create variation in daughter cell, but meiosis can. There are 2 important steps, or we say 2 important process or event in the meiosis that guarantee variation. So meiosis have 2 phases, 2 division phases, unlike in mitosis which has only 1. So here meiosis 1 consists of prophase 1, metaphase 1, anaphase 1, and telophase 1. At the end of telophase 1 is already half the chromosome number from 2N into N. While meiosis 2, you have prophase 2, metaphase 2, anaphase 2, and telophase 2. At the end of meiosis 2, the daughter cell remain as haploid. So in the interphase is exactly same like what happened in mitosis. You also have S phase, which is a cell division, which is a chromosome duplication. So one chromosome now it duplicates, so become 2 chromatic attached together at the centromere. This is still 1 chromosome, but this is 1 chromatic, this is another chromatic. And why it's called sister chromatic? Because this chromatic and this chromatic, they are genetically identical to one another. So that's why we call them as sister chromatic. Now let's proceed for meiosis 1. We start with prophase 1. In the prophase 1, the chromatin again condense to form chromosome. So now, there's one important event called pairing of homologous chromosome. What is the meaning of homologous chromosome? Homologous chromosome, the term homo means same. Homologous chromosome referring to those chromosomes having same length. For example, can you tell me now how many chromosomes you have in this cell? This is 1 chromosome. If you look at my arrow, this is another chromosome. This is another chromosome. This is another chromosome. So total you have 4 chromosomes in this cell. So out of 4 chromosomes, 2 pieces of chromosome they are short, 2 pieces of chromosome they are long. So 2 short chromosomes pair together to form 1 homologous pair. The other 2 long chromosomes they pair together form 1 homologous pair. So this is referring to pairing a homologous chromosome, which means those chromosomes having the same length, they'll pair together to form homologous pair. This is not seen in mitosis. Then when the homologous chromosome they pair together, what is the purpose of pairing? Because they want to exchange chromatic arm. This process is called crossing over. As you can see, this is a homologous pair as they have the same chromosome length. So during the crossing over, they will exchange their chromatic arm. This process, the point where they exchange their chromatic arm is called chiasma. Plural is called chiasmata. After they exchange, they overlap their chromatic arm, they'll exchange the chromatic arm. From here, this chromosome carrying a little bit of feature of the other blue chromosome. Well, this blue color chromosome carry a little bit of feature from the this yellow chromosome. So here it creates variation in the gamete. That's why sometimes you can have your look exactly like your father, but you have your mother's eyes. That's how the crossing over provide variation in the product form. So in this process, the crossing over involve the action of chromatic arm between non-sister chromatic. What is non-sister chromatic? This chromatic in yellow and this chromatic in yellow, they are sister chromatic because they duplicated from the same chromosome. And this one in blue and this one in blue, they are also sister chromatic. But then this one chromatic in yellow and this chromatic in blue, they are non-sister chromatic because they are from different parents. So crossing over involve overlapping action of chromatic arm between non-sister chromatic so that they can create variation in the zygote, in the gamete. And then in the prophase 1 later on, the nuclear membrane and nucleus start to disappear, spindle fibers start to form. Now they start to attach to the chromosome. In the metaphase 2, the homologous chromosome is aligned at the metaphase plate. Look carefully. In the mitosis, it is chromosome aligned at metaphase plate. But in this meiosis, it is homologous chromosome aligned at metaphase plate. Later on, when you proceed to anaphase 2, then this chromosome will be pulled to the opposite arm. So you can see this chromosome and this chromosome will be moving to the opposite arm, and then it start to break apart. And then when you proceed to the telophase 1, the chromosome will start to form compact at the upper. So now the two daughter cells is having half the chromosome number. Because if you remember, I asked you at the beginning, here how many chromosome? 4 chromosome. But when you reach the telophase 1, you have only 2 chromosome. This is 1 chromosome, this is 2 chromosome. This is 1 chromosome, this is 2 chromosome. Each daughter cell have only 2 chromosome. So half from the parent cell. So from here, the telophase 1, it already formed different chromosome number. And one of the important feature is happening at metaphase 1, which is called independent assortment. If you see, this is one chromosome, this is the other chromosome. So the chromosome in blue, dark blue, and the light blue is arranged in this way. This one in light blue and the dark blue is arranged in this way. What is happening is the arrangement can be in the other way around. The dark blue and the dark blue together in one line. The light blue and the light blue together in one line. The different arrangement of homologous chromosome can also create variation in the daughter cell form. After the telophase 1, the cell will proceed for meiosis 2. In the meiosis 2, start with prophase 1. Prophase 1 is quite similar to the meiosis 2 is quite similar to the mitosis process. So here, the centriole will replicate again, and the centrioles and the nuclear envelope will start to resolve, and the spindle fiber will form attached to the chromosome. Now, in the metaphase 2, the chromosome will align at metaphase plate, and then they will be pulled to opposite end by the spindle fiber. The process of pulling, again, always remember, is leading by centromere. Now, when it reach the end, then they'll start to form new daughter cell. So it formed by telophase 2. So this is again same. Proceed to cytokinesis, formation of spindle formation of this cleavage furrow to pinch the daughter cell into 2. So at the end, the daughter cell form is haploid. So from overall, initially you have only 1 parent cell in meiosis 1. Now you form 4 haploid daughter cells in meiosis 2. This is how meiosis have the chromosome number. This is a picture viewing the actual division in the human cell. So this is a comparison between mitosis and meiosis. It's very important for you to know. So you can see, for example, here, what is the diploid chromosome number of this original parent cell? The answer is 6. Why? Because you have 6 pieces of chromosome. This is 1, 2, 3, 4, 5, and 6. So the 2n is 6. So This is the parent cell. So how many chromosome number? Still 6. 1, 2, 3, 4, 5, 6. Just that each chromosome is having 2 chromatic. So in the meiosis, how many chromosome I have? Still 6. 1, 2, 3, 4, 5, 6. So notice that the differences is in the meiosis, the prophase one you have pairing of homologous chromosome, but then in mitosis there is no pairing of homologous chromosome. When we proceed for metaphase, in the metaphase of mitosis, chromosome align at metaphase plate, but then in metaphase 1 is homologous chromosome align at metaphase plate. In the mitosis, at the end you form 2 daughter cells, which is diploid and genetically identical to the parent, or having 6 chromosomes. 1, 2, 3, 4, 5, 6, seem like the parent. However, in the meiosis, you can see at the end of meiosis 1 is already half the chromosome number. In this style, you have 1, 2, 3 only. Here also 1, 2, 3 only. So it's already half the chromosome number. So at the end of meiosis 2, it produce only it produces 4 daughter cells or half the chromosome number from the parent. So initially here the parent it have 6 chromosomes, but now is having 3 chromosomes only in the daughter cell form. This is 1, this is 2, this is 3. Only 3 chromosomes is formed. And I want you to notice the formation of variation. Do you notice that there is crossing over? So the exchange of chromatic arm, so that's why the daughter cell form is having variation different from the parent. And the other one is the independent absorbment I told you. If you look at this picture, they can be see the independent absorbment clearly at this picture. So if you see here, the chromosome in red, they align. How they align? This is red chromosome, this is blue chromosome, this is blue chromosome. So it's red, blue, blue arranged in one line. And in this part, this is blue chromosome, this is red chromosome, this is red chromosome. So the arrangement is blue, red, red. So later on, the daughter cell form also have blue, red, red. Well, in this one is red, blue, blue. So at the end, the daughter cell form is having red, blue, blue chromosome. In case now, imagine if the arrangement is red here, and red here. So all red in one line, blue here, blue chromosome here, blue chromosome here, all blue in one line. So what is the result? If all blue in one line on your right, so all the daughter cell form will all blue. Same thing on your left. If all red in one line, what happened to your daughter cell form? The daughter cell form also all red only. So the homologous chromosome can freely arrange, either they want in some have red, some have blue, or no. So this also create variation in the daughter cell form. This picture also showing the differences between mitosis and meiosis. Mitosis happen in somatic cell, which is the body cell, except gamete, but meiosis happen only in gamete, which is sperm and ovum. So mitosis, one parent cell produce 2 daughter cells, while meiosis, one parent cell produce 4 daughter cells. However, in the meiosis, one parent cell, which is diploid, produce 4 haploid cells, but mitosis still a diploid daughter cell. You look at the division of chromosome number. In the mitosis, the chromosome number is half, but in the meiosis, the mitosis, the chromosome number remains same as a parent, but in the meiosis, the chromosome number is the DNA number is half from the parent. There's no pairing of homologous chromosome in mitosis, but in meiosis, there's pairing of homologous chromosome. There's no crossing over in mitosis, but meiosis have crossing over. And the centromere is divided at the anaphase, but there in the meiosis, the centromere do not divide at anaphase 1, but it divide at anaphase 2. So mitosis is a conservative process because the daughter cell genotype identical to the parent, but the meiosis promote variation. So the mitosis, the cell undergoing mitosis can be either diploid or haploid, which means if the parent is diploid, the daughter cell form will also diploid. If in mitosis, the parent cell is haploid, the daughter cell form also haploid. But in the meiosis, the parent cell definitely is diploid. The product will be haploid.